So I'm going to talk a lot about what I do as a research soil scientist with the Agricultural Research Service, which is the research arm, sort of the sister agency of the NRCS. Now, just to give you a little bit of perspective on where I'm coming from as a soil scientist, this is sort of how I look at things. So I'm focused on what's happening below ground, but obviously what's happening below ground has a lot to do with what's happening above ground. And I've been a bench chemist, you might say, for 27, almost 28 years. I started doing lab work when I was a senior in high school, so you can do the math and figure out how old I am. But what I do is try to come up with lab methods and ways of interpreting lab data so that we can understand soil health. And you guys that are here in the audience, I consider you the end user of what I do. And so I'm really excited to come to producer workshops and interact with you. So soil health has been in the news a lot lately. I think you all know about the new NRCS Soil Health Division, and they have soil health specialists spread across the country now. And they're really gearing up for a soil health assessment on a nationwide level. There's also the Soil Renaissance, which turned into the Soil Health Institute. And that was with the Farm Foundation and some other agencies. There's the Soil Health Partnership, that's National Corn Growers. There's the Foundation for Food and Agriculture Research. They have an initiative on, I think it's called Healthy Soils and Thriving Farms. And then the Nature Conservancy has a soil health initiative, and there are a lot more that you can find in the news. So really, what is soil health? And if you start looking at definitions for soil health, they're usually pretty broad. And this is the one that I like to use, but the NRCS has a definition, the FAO has a definition, but they all kind of come back to the same basic principles. And that is the function of the soil system, and then it includes something usually focused on the biological activity or the biological productivity of the soil. And we want to focus on soil function, and sometimes people say, well, what is soil function? Well, soil functions are the things that the soil is doing that we care about, the services that the soil is providing for us. And then the reason we're focused on biological measurements in soil or biological productivity is because even though we may have known for a long time that soil biology was important, we didn't necessarily have the tools to measure soil biology, and now we do. And we're developing new methods all the time. And so we're trying to bring that together with the traditional approach of measuring soil quality. So this is my interpretation of the history of soil health. It all came out of soil conservation, and we all know about the Dust Bowl in the 1930s. And then in 1935, Congress passed this public law, and they established the Soil Erosion Service, which later became the Soil Conservation Service, and is now the NRCS. Then in the 40s, there was a book called Plowman's Folly, and there were other writings that came out during that time that criticized intensive farming practices. And then in the 50s through the 70s, there was a lot of pressure to conserve soil and water resources. And again, that was really focused on keeping soil on the landscape, reducing erosion. So it was all about the quantity of the soil and keeping the soil in place. And then in the 1990s, people really started researching soil quality and what we now call soil health. And that was looking at the characteristics of the soil, not just the amount of soil. But at the same time, there were several arguments that kind of came out against the soil health concept, because at the time, it really was just that, it was a concept. People were concerned about the fact that maybe this is too qualitative and not quantitative enough. It's just too descriptive. We can't, wrap our minds around it. What are we talking about here? And we know that soil use varies. So if we're not using the soil for the same thing, then how does that factor into your assessment of soil health? We also know that soils are not the same. On one field, the soil may have certain characteristics down the road or in the next state. Soils may be very different. So how do you factor that in? And then soil function, going back to the services that the soil is providing for us. There are many different soil functions, and so which one do you choose? What do you emphasize? What is it that you're trying to optimize? That third line there, measurable and interpretable, those are the things that I work on a lot. So we have to be able to measure it, right? And as a laboratory scientist, I spend a lot of time trying to figure out how to best measure things. And then interpretable. So if we measure it, that's fine, but if we can't interpret it, it's not very useful. And then the last issue, anytime that you're doing something new, there are always socioeconomic and then regulatory concerns that people have. You know, where is this all going? So it's my job to take soil health from a concept to a science, and I'm building on several decades of research that other scientists and a lot of ARS scientists have been doing since the 1990s. So how does this work? Well, like I said, we have our soil functions, and we want to select those things that we can measure in the soil, and we break those into the three basic categories: the chemical, the biological, and the physical. And we pick these things because these measurements because they relate back to those ecosystem services or soil functions. And some examples would be soil conservation, soil productivity, and then a host of different environmental protection services that the soil provides. This is an example of one study that I conducted with some of my colleagues back in 2008. And we measured everything under the sun just because we can. But you can see where we have a breakdown of the biological, chemical, and physical soil properties. Now, a lot of people want to know what's the difference between soil health and soil fertility. And I say soil fertility is a part of soil health. It's a very integral part of soil function and soil productivity. And so we're not throwing the baby out with the bathwater, so to speak. So if you see on this list of some soil health measurements, and these are measurements that I commonly use in my lab, in my assessments, the ones I circled in red are the ones that you're probably the most familiar with. Those are the traditional measurements that you might get from a fertility lab. So it includes things like phosphorus, potassium, and pH, and then you may be familiar with organic matter. But then what we've done is we've expanded upon that. We now have some new physical measurements, as well as a pretty wide range of soil biological measurements. So if we're going to develop a soil health assessment, these are the basic steps that we're going through. And this is a process. This is a process that we're involved in now. We're still working on this. So this is not an end product yet. So the first thing we have to do as scientists is pick those soil functions and then the lab measurements that we think link back to those functions. And then the second step is to select the laboratory method. And when I say this, people kind of roll their eyes or like, yeah, whatever. Who cares about the lab method? And since that's what I do, I focus on that quite a bit. And I'm sure many of you have sent soil samples off for standard fertility testing. And you may not think at all about the method that's behind that, but methods vary. So how many of you, it's kind of hard for me to see, but how many of you have sent soils off for standard fertility testing? Most of you, right. Have you ever sent samples to different labs at the same time? Yeah. And so some people will notice that maybe the results are not the same. And in large part, that's often due to differences in the methodology. So the methods matter, right? And then after we have the methods, then we have to figure out how those laboratory values actually relate back to soil function. So we have to quantify that. We can just assume that more is better, right? But how much better? And there's the law of diminishing returns. So if you have 3% organic matter, is that enough? Do you need 10%? I mean, where are those thresholds, right? And then the last thing, which is sometimes overlooked, but we need to quantify the effects of management and how management affects those measurements so that we can provide recommendations. Because we can give you all the data in the world, but if we can't tell you what it means and then what to do about it, again, it's not very useful. So to start with that first step on soil functions, this is a list of some broad soil functions. And from the soil health perspective, you really have to accept that all of these things are important. You know, as producers, number 5, crop productivity is probably at the top of your list. But if you look at number 1, that's water flow and retention. You know, that relates to crop productivity, but it's also important for other reasons. Physical stability, nutrient retention and cycling, biodiversity, and environmental remediation. So these are all soil functions that as soil health scientists we're trying to bring together. Okay, well, my picture's a little big there, but this is just another example of soil functions, but looking at it from a process-oriented perspective. So you can kind of break these things into categories like chemical reactivity, nutrient availability, general biological activity and substrate for the microorganisms in the soil, water partitioning, organic nitrogen, and then the microbial community. And I'll talk a little bit more about some of these categories later. So what is a good soil health indicator? So how do we pick those indicators? Well, it needs to be meaningful, right? And it has to reflect soil function. So I've said that a few times already. But then you also need to think about sensitivity. So if we're measuring something and after 10 or 20 years of improved management and you switched to no-till, you added cover crops, you're doing all these things, and 10 or 20 years later you still don't see a change in this particular measurement, then maybe it's not a very useful soil health indicator. On the flip side of that, you can have an indicator that varies really rapidly based on just changes in daily temperature or rainfall. So that might be too sensitive to environmental conditions. So you want to find something that's going to be sensitive to the changes and the improvements that producers are making on their land. So if you're looking at producers, you might say, 3 to 5 years. I mean, as producers, if you're sending off, let's say you decide to switch your management today. You decide to go to reduced tillage, and then you add cover crops. If you're sending in samples to a soil health lab, how quickly do you want to see a change? I mean, I suppose we could all say, I want to see it now, but what do you think is reasonable? Let's throw some numbers out. 2 years? 5 years? 5? Anybody else have an opinion? 3? 4? Okay, well, and so typically we're shooting for something that's going to be sensitive to maybe 3 to 5 years change in management. Now, obviously, there are a lot of other factors that are involved there. It depends on your soil type, it depends on where you started, where you're going. But that would be, I think, an ideal time frame. And then, of course, we have to make sure that it's something we can really measure and that it's affordable. I'll talk a little bit more about that, and then interpretable. So when we select methods, and I'm talking about methods that we're hoping ultimately a service lab is going to make available to producers like yourselves, right? Not talking about some of the fancy stuff that we do on the research side. But you have to think about the equipment costs and the required skill level. So, it doesn't make much sense to recommend a method that's going to require, a $20,000 GC or a $200,000 mass spec, right? And then if the skill level required to actually go through that process in the lab, if you have to hire somebody with like a PhD in phase extractions or something like that, it may not be really feasible. And then all the consumables and the labor that go into that method. And at the end of the day, there's a trade-off. You have to balance the cost of measuring that with the interpretability. What are you getting out of it? Like, what is that end product? And what can you do with that information once you have it? So these are just some general costs on soil health analyses, and this is not from any particular lab. I just put this together from my own experience. There are several labs across the country that offer these tests, and you might come across different prices. But my point is that you start adding all these together, it starts adding up pretty quickly. So when you guys send in your soil samples, whether it's for fertility testing or for soil health analyses, who's paying for that? Is somebody paying for your testing? Or are you? Yeah, so you are, right. And I know there are some programs out there, a lot of programs out there now. Missouri, where I'm from, there are a lot of state programs and some federal programs that are either partially or totally subsidizing soil health testing. But who funds those programs? Right, you do. So when I talk about budget and cost, some of my colleagues that are more on the research side, maybe in the university, that are not necessarily in a position like mine, roll their eyes at me when I start talking about some of these tests that I'm evaluating in the lab that are cheaper than something really fancy and sophisticated. But we have to keep this in mind, right? So if we go just through some of these tests, and I'm sure other speakers have talked a lot about some of these things, but basic chemical reactivity, nutrient availability, these are the things we're the most familiar with, right? Then we also get into some of the physical properties, and we call that physical, and water partitioning is one of the primary things that we're concerned about when we talk about soil physical properties. Not the only thing, but it's really important. And so water partitioning is just when the rain falls, where is it going? So you're partitioning it. Is it running off? Is it going into the soil? Where is it going? And a lot of that is dictated by soil properties such as aggregate stability, bulk density, soil texture, things like this. On the soil microbiology side, we have potentially up to 1 billion soil microbes just in 1 tablespoon of soil, and that's a lot. And when I give talks to high school students, I play this little game with them where I ask them to see how high they can count in their heads for 30 seconds. And they can usually, the fastest one can usually get up to maybe 150 or something like that. And then I tell them that, okay, so if you were counting 1 billion soil microbes at that rate, it would take you about 6.5 years to get to 1 billion. And that would be with no breaks and no recess, nothing like that. And I go through that exercise just because sometimes it's difficult for us to wrap our minds around what 1 billion really is. It's a huge number. And so in the soil, we have all these microbes. There's bacteria, fungi, yeast, protozoa, algae, nematodes, all kinds of things. So why do we care about all of these microbes? And it's primarily because the microbes are the ones that are responding very rapidly to changes in their environmental condition. So they're the little harbingers of what's happening, and they're very good indicators, very good at indicating rapid changes in soil. So the chemistry and the physics of the soil provide the context, and that's the habitat in which the microbes are living. So when I'm approaching measurements for the soil biology, this is how I think about it. I break it up into these 3 categories. So what are the questions that we want to know? Well, we usually start with how many. So you can think of that as like a population or a census. How many microbes are there? And so we can do that by measuring something called microbial biomass, or we can use total phospholipid fatty acids. Those are 2 of the most common ones. But once we have a handle on how many microbes are there, then we want to know who's there. So who are they? So I can go through and I can count the number of people in this room, but maybe I want to know a little bit more about the demographics. How many of you are raising different kinds of crops, or how many of you are strip-tillers versus something else, right? So we want to know more about who's there. We call that microbial community structure, and we can get at that by looking at the soil microbial DNA. So we can actually pull the microbial DNA out of the soil and sequence it and identify the organisms that way. We can also do what I mentioned earlier, that phospholipid fatty acid analysis. And I know of at least 2 service labs across the country right now that are offering that to producers. And with that, you don't get something that's species specific, but you get some broad microbial categories. And from that, you can infer something about the microbes that are there. This last one, this is the one that I'm the most interested in. This is what I spent most of my time thinking about. And that is, what are they doing? So just because the microbes have the genetic potential to do something, so the DNA is there. It doesn't mean they're actually doing it, right? So these genes get turned on and off, and their activity levels change. So we want to know, what are those guys really up to in the soil? And that's where we start measuring enzymes, things like soil respiration. So if you're thinking about, well, hold on, I'll get to that in a minute. So if we're interested then in the general biological activity in the carbon food source, the things that relate to that we measure: organic matter, soil respiration, active carbon, particulate organic matter, enzymes. So I threw all these up here because I think you're probably going to start seeing more of these kinds of indicators or analyses available through different labs. I already know of several that are offering some, if not all of these. But soil respiration is an important one. So again, I can count all of you that are in the room, and I could measure your activity level right now. And it might be low because you're sitting and it's right before lunch. But then if I asked you all to stand up and start doing jumping jacks, would you all do it? I don't know. But your activity level would change. So the population count has stayed the same. The community structure, the genetic potential has stayed the same, but now all of a sudden your activity level is very different. So that's why we are interested in that. And microbial enzymes is another way to look at microbial activity. So there are all these enzymes that we can measure, and they all relate back to different nutrients and nutrient cycles. So if we're interested in phosphorus, we might measure phosphatase, aryl sulfatase for sulfur, things like that. For organic nitrogen supply, we look at potentially mineralizable nitrogen. And this one's kind of tricky. So we know that organic matter in the soil has the potential to supply nitrogen to plants during the growing season. And what we'd all really like to be able to do someday is to measure that and then use it as a credit against our fertilization, our nitrogen application. But the difficulty we run into there is that the timing of when the microbes are releasing it in the soil and mineralizing it and making it plant available is not necessarily when the plant wants it. So we haven't really been able to achieve that yet, but maybe someday. So for microbial community structure and diversity, like I said, we have DNA, we have the phospholipid fatty acids. That gives you the broader groups. We can also do things like pathogen bioassays. But the key here then is the cost and the interpretation. And so costs on things like DNA and sequencing are rapidly coming down, but they're still pretty high up there. So just to give you an example, this is some work that I did about 10 years ago on microbial community structure, and this is phospholipid fatty acid data. So you don't need to know all the details of these different bars, but I'll explain that the height of the bar is essentially an estimate of the total population. So it's all the PLFA markers that were in that sample. And then there's a list of categories on the side, and those are the different microbial groups, and it sums up to that total. So if you look at this chart, on the far left, that really tall bar is Tucker Prairie. So that's a native, never plowed prairie. It was grazed for a few years back in the 1950s, but essentially it's what we call our virgin prairie. Then the next one is CRP, followed by a no-till corn, soybean, wheat rotation with cover crops. So that's our most diversified row cropping system. And then we have some mulch till, and it goes down to no-till with continuous corn all the way down. The lowest one is conventional tillage, continuous corn with no amendments of any kind for 125 years, something like that. A lot of these samples came from Sanborn Field. So the trend to notice here is that the total population of microbes has essentially exponentially declined as you increase the disturbance and reduce that crop diversity. But the problem we still run into is how do we interpret these results? How do we put this data into context so that if a producer is going to get a test, what is that really telling them? And so we're still collecting a lot of data. We're in the process of measuring this under a wide range of management practices and soil types, and looking to see if we can extract some really useful information out of this. Now, I'll go back to this slide real quick. So all those different categories that are on your right, it's probably hard to tell in looking at this graph, but all of those categories are present in every single one of the samples. So from a very basic approach of microbial diversity, all of these different treatments are equally diverse, but that's because we just have these broad categories. If you really knew the species composition, there's probably a big difference between what you see at Tucker Prairie and what's down here in this conventionally tilled continuous corn plot, but I can't pull that out of this data. So when we're sampling for soil health analysis, and you guys are familiar with, you think these things through before you collect your samples, right? So, why are you sampling? And that's going to dictate a lot of what you do with your soil sampling. So when are you going to collect your sample? Because with soil biology, there is a stronger impact of that temporal variability or these seasonal changes, right? So if you're sampling in the fall versus the spring or in the middle of the growing season, that can have an effect. Where are you sampling? So there's an issue with spatial variability that's always been an issue, but in particular for soil biology, you really want to think about where you're sampling within a field and the management. So if you have different management imposed, you don't want to necessarily mix those samples, and then depth. And then how are you sampling? So with some of these soil biological measurements, we have to be really careful about how that soil sample is handled after you pull it out of the ground. If you're going to measure phospholipid fatty acids, you have to send that sample to the lab overnight. You can't let it sit around, on the dashboard of your truck or on your kitchen counter for a week and then send it off. Some of the work that I've done, I've found that just letting a sample sit for 2 weeks at room temperature in a bag, still moist, you can lose half of the fungal markers and 30%, 10 to 20, 30% of the other bacterial markers. So that's a big deal. And it's also on the lab side a consideration that we have to think about if we're wanting to recommend a method for a service lab. Because how realistic is it that a producer is going to be able to collect a sample and get it to the lab in a timely fashion? So we try to pick methods that are more robust to some of the handling effects that they might experience. And so for field scale variability, you look at this is actually an EC map, an electrical conductivity map, but it just shows that within one field you can have dramatic differences from one place to another. And so if you're sampling and you collect a sample in that red zone one year and then you come back and collect your sample in the blue zone the next year, you may see a big difference. And is that really due to your management, or is that just due to where you decided to collect your sample? So you want to think about the landscape. You want to think about if you have erosional areas or depositional areas. And then think about what your question is. What is it you really want to know about that field? And then pick a place that you're going to sample and then stick with it. And I said depth was important, and that's particularly important with soil biology because the organic matter is concentrated near the surface, and that's where all the soil microbes are. That's where the activity is. That surface layer, that's the interface with the atmosphere. So if you're collecting a sample of 0 to 6 inches, you don't want to compare that to a sample that you collected 0 to 12 inches because, again, that difference may just be due to the depth of sampling and not really answering the question that you want to answer. The rhizosphere, that's the soil that's right there next to the plant root. Okay, and if you know a lot about soil microbes, you know that they're getting a lot of their food from the plant roots. So the roots are exuding all these carbohydrates into the soil. The microbes, it's like the buffet line, they come up and they're consuming that. So where do you find most of the soil microbes? Right there next to the plant root. So again, if you're doing a soil sampling, and you sample right next to a plant where you're capturing the rhizosphere soil, and then you sample like between rows where there are no plants, again, you might see a big difference just in those samples. So you got to think about those things. So how do we interpret all of this soil health data? So what we want to do ultimately is to be able to take the lab data and convert it into something like a grade or a score, something that's more interpretable. Because if you send me a sample and I measure glucosidase activity and I come back and say, hey, your sample had 200 micromoles of PNP per gram per hour and yours had 30, what does that really mean? And you have to be able to interpret this lab data. And then if you can interpret it, then it's going to help you prioritize your management decisions. So that's why we want to do this. The index that I like to use the most is called the Soil Management Assessment Framework. And this was developed going back to the 90s by several ARS scientists and some of their colleagues. And it takes these biological, physical, and chemical lab measurements, and it transforms them, it scores them. So it converts them into a number, like a percentage between 0 and 100%. And what that means is if you get a score of 100%, then your soil is functioning at 100% of its potential for that particular indicator and the function that it relates to. But if you get a score of 50%, what does that tell you? There's room for improvement, right? So within this particular index, we have on the biological side, we have organic carbon, mineralizable nitrogen, glucosidase activity, microbial biomass. Physical side, we can do bulk density, aggregate stability. And on the chemical and nutrient side, we have the standard things like pH, P and K, sodium absorption ratio if you're out west, and that's important to you. But this is what I really like about the Soil Management Assessment Framework. And it's the only index that comprehensively accounts for soil taxonomy, texture, slope, and climate on the front end. So at the beginning of my talk, I was saying something about how we know that soils differ from each other. So soils have some inherent differences, and so you can't expect one soil to necessarily do as well as another soil, like an alpha sol versus a mollisol. So the SMAF, as we call it, Soil Management Assessment Framework, takes this information, all this site-specific information, the soil taxonomy, the texture, all of that, puts it in on the front end, and then you add the lab data and it scores it. So you're accounting for all those inherent differences up front. Now, I will say that the Cornell Soil Health Assessment was based on this particular approach, and so they regionalized it for New York and their area, and then recently they've expanded that to include more states towards the Midwest. But let's see. There's an example of where I used this SMAF in Missouri. So these are a bunch of different management systems on the Missouri clay pan. And you can see, and you don't need to worry about all the different management acronyms at the bottom. What you want to pay attention to are the general categories I have up here in red. So across all of these management systems, there was a clear break between the perennial and the annual cropping system. So everything on the left is the perennial and the right is annual. And this is, again, these are the scores, integrated scores. So then if you look within the perennial systems, we see a break between the cool season grasses and the warm season grasses. And this usually raises a lot of questions. Why would the cool season grasses score higher than the warm season grasses? Particularly when the warm season grasses are considered the native grasses in our area. And some of that has to do with the fact that if you think about cool season grasses and their root architecture and rooting density near the surface, they have more roots near the surface. And with the warm season grasses, they tend to be clump forming, there's more exposed soil. So from an ecological perspective, you might argue that, that we shouldn't score the warm season grasses lower than the cool season grasses. That's something we have to address in the future when we start thinking about native ecosystems. But then if we look over on the annual cropping system side, you can see a break with increased rotation diversity and reduced tillage scoring higher. And then as you increase the disturbance and reduce the diversity, then the scores drop. We did a similar study on Sanborn Field and Tucker Prairie, and these are the same systems that I showed you earlier with the PLFA data. And similar to that clay pan study, we had a break between the perennial and the annual cropping systems. So we saw the same thing. And then if you were to look down through all of these different management systems, you'd see the same trend. As you increase disturbance and reduce diversity, and you move down to those conventional tillage practices with monocultures, then your soil health scores rapidly decline. So what I did is I took several of these studies that we've done in Missouri, and I put them together into what I call the agricultural continuum of soil health, from higher to lower, because really it is a continuum, right? And I put the breaks between perennial and annual cropping systems, and then within that, the different management practices that tend to dictate whether you have higher or lower scores. Now, obviously, there are several factors that go into this, but I just wanted to illustrate that within perennial systems, the things that tend to dictate your soil health scores there would be the vegetation type. So what is the vegetation? And then are you removing that vegetation? So biomass removal, harvesting is going to pull some of the nutrients out, right? And it's going to end up reducing some of those scores. And then on the annual cropping system side, that's that same pattern that's dictated by disturbance and crop diversity. So on the regional and national research side, there are a lot of initiatives in place now to come up with both regional and national soil health assessments. So I use the SMAF, but there are still a lot of gaps in the data in order to make that fully comprehensive. And there are some additional indicators and lab measurements that we want to add to that to make it also more comprehensive. So we're in the process now of looking at different methods and protocols, things that we can add to that assessment. And in order to do that, one of the things we need to do is develop regional databases. So we really understand what the range of soils are within an area and what the potential is in that area for those different soils. And then the third thing I have here, it's the really important part to me. It's also the hardest part of this whole process, and that's taking those measured values and actually quantitatively linking that back to soil function so that it's outcome-based, right? Those processes and services that the soil is providing. And that's the hard part. But once you've quantified that relationship, then you don't have to worry about that part anymore. Then you just measure the lab data, right? And then you can do this last thing, which is interpret that for management decisions, which is ultimately what we're really trying to do. And this is one of my favorite quotes. And I will take any questions anyone has. No questions? Or if there are, I can't see. Yes?
Oh, hold on. We have a mic coming, so.
On the perennial prairie systems, where you're showing the diversity of that wasn't as great as, some of the other systems that you had up there with the cooler grasses. Mm-hmm. Do you think that's possibly, is animal impact a part of that perennial system, or is that just without animal impact?
Well, in those systems, we didn't have any livestock incorporated into those management systems, but that is a good point. Obviously, one of our goals is in these studies is to include more diversified integrated management systems to see how these management practices interact with each other. So we can go in and well, for example, one of the questions I often get on that no-till corn, soybean, wheat with a cover crop system, people sometimes ask me, well, how much of that improvement is due to the no-till and how much of that improvement is due to the cover crop? Well, it's not that simple because those practices are interacting with each other. So it's like the classic saying that the whole is greater than the sum of the parts, right? And so I think that's a very good point that livestock can have an impact on what you see in the data.
Over here.
So I've seen different sets of these soil health tests, and very few, go back and relate it to like a yield of a corn and soybeans. And what I have seen is a little confusing. And it might be just in the testing and maybe the time of those practices have not been in there long enough. So anyway, my main question is the one you showed, the different systems with the different soil health scores. Are you, and maybe I missed this, are you guys going to do yields checks on those different?
Right. So that's one of our goals, at least for me and my colleagues over the next few years, is to pull together not only yield data, but also water quality data, and essentially verify or validate the scoring system. And in some cases, we'll probably have to modify the algorithms that we're using. But we still have a lot of work to do on that end. So I will say we need to get more yield data, and we need to pair it with the soil health data. And we need, again, to make this outcome-based, we have to quantify that relationship. And so we're still working on that. Well, we often do see a trend with yield that relates to improved soil health for several potential reasons. Again, as we increase organic matter, there's more of an organic nitrogen pool available. We see improved soil structure, and so we have better water storing capacity. So there are a lot of things that we know just sort of empirically about what's happening as we improve soil health. But pulling together the data is something that we're trying to do now. And I can give you one example of where you see the opposite. Like, for example, we have a set of experiments that are continuous corn all the way to continuous soybean in every rotation possibility, essentially in between. And with continuous corn, some of those soil health indicators are going to increase because of the increased residue and all this biomass that you're returning because it was a no-till system. But you still see that 10 to 15% yield decline that you get with continuous corn, right? So in that case, you have sort of the opposite effect happening. So there are some things that still need to be looked at and worked out and figured out in terms of how to interpret the soil health data.
One over here.
Do you envision a time when you can determine the number and the type of microbes and how that might influence how we, what and how much inputs we put into a crop?
Well, I would think that eventually that will be a factor that we use to make that decision. And I don't want to sound like a broken record. A lot of this is ongoing research. So again, we have several projects just with my unit and my colleagues where we're looking at the metagenomics of the soil microbes. And we're also collecting yield information and a bunch of other soil information to hopefully get to the point. I think just from a perspective of like basic science, it makes sense. We know that the microbes are involved in all these different nutrient cycles. We know that they're involved in creating and maintaining soil structure. So they're an important part of that system. We just haven't figured out how to quantify that, and we haven't established that relationship yet, but I think we will.
To be able to have a consistent and representative sample for lab testing, based on what you mentioned, what would be your recommendation as far as when and which depth to sample?
Well, so it comes back to the question that you're wanting to answer. If you can afford to collect several samples within a field, then I would make sure that so if you're farming a field, you know that field, right? You know there are areas that have maybe low production versus others that have higher production. So if it were me, right, I would go out and I would want to get samples from both of those contrasting areas, because I would want to see what is it about this area that's not producing that makes it different from the area that is producing, because that helps you decide what to do management-wise. Now, as far as the depth, we typically sample 0 to 6, and so, that's the part of the soil that I really do think, from a crop perspective, is the most important. Obviously, I work in the clay pan, so that subsurface layer plays a big role in the hydrology of the system, and it's important, but it's not changing as much as that top 6 inches, and that's the part where the crop roots are going to be and where the microbes are going to be. And so I don't see any reason to change that. And then, if you can't afford to send in several samples and you want to do a composite sample for one field, then you can do that. It kind of depends on your budget. And then in terms of the timing, so a lot of our research is done in the fall just because it's convenient to get out in the field. But again, down the road, the way I look at it, I would like to see soil health testing be part of, incorporated in with standard fertility testing. And so if you, as a producer, you're going into your growing season, you want to know what's going on in your soil so you can plan for that growing season. And so I don't want to tell producers they have to go out and sample multiple times a year. It'd be nice if they just go out in the spring, get their sample, get all their information from that, and then go from there. Now, if you're not a producer and you're an ecologist and you want to do a different sort of soil health assessment on like native prairie or something like that, then you have a completely different approach that you might take. But I'm just talking about from a producer's perspective. That's what I think would make the most sense ultimately.
Hi, I'm curious about the research that you have said that you're doing to correlate the yields with the soil health sampling. Is the research being done nationwide? Like, are all the ARS agencies working together to work on collecting this data nationwide? And is there farm-scale research that's being done in a real farm situation and not just on the research plots?
Right. Well, there are a lot of initiatives right now to bring together ongoing research. So if you look, like within the ARS, a lot of locations have had their own research projects for many decades. Ours has been going on since 1991. And we have some networks. There's the long-term agro-ecosystem research network. So we're talking to people within NRCS and the Soil Health Division to pull that data together and to use that for the nationwide soil health assessment. And then as far as on-farm research, we do some of that, like I've done some of that, but the Soil Health Partnership is a good example. That's the National Corn Growers, and Nick Gazer and I worked with them, and a lot of ARS scientists are in the advisory board. And that covers, I think, 10 states now and over 100 different producers, and these are replicated strip trials. And of course, it's on-farm data, so it's subject to all the caveats that come with each farm is being managed by a different person, and things sometimes go wrong. But we've been looking at the data, preliminary data, and what I like about that on-farm approach is that you can really get management systems and soils that you wouldn't necessarily be able to access otherwise. And it shows you a lot of the broad relationships and the broad factors that are controlling what you see at the field scale. So it helps put everything in context. So I think both types of experiments are necessary, and they both bring something different to the table in terms of the information that you can get from that.